Hello students, welcome to readmedprep Academy channel. Today in Molecular Genetics Part 5, we shall discuss about transcription. According to Francis Crick, who proposed the central dogma of protein synthesis in molecular biology that states that genetic information flows from DNA to the cellular action. DNA undergoes transcription resulting in the formation of RNA, and this RNA undergoes translation to form proteins, and these proteins are responsible for cellular action. What is transcription? The process of copying genetic information from one strand of DNA into RNA is termed transcription. Here you can see that the DNA is transcribed into a messenger RNA. Normally, in a DNA molecule, adenine pairs with thymine and thymine pairs with adenine, whereas in the synthesis of the messenger RNA from the template DNA, the uracil is formed instead of thymine. That is the only difference between the DNA and RNA. That is where the DNA strand contains thymine as the nitrogenous base, whereas the messenger RNA or the RNA contains uracil instead of the thymine. What is required for transcription? This process of transcription takes place in the presence of the enzyme DNA-dependent RNA polymerase, and the strand on which the messenger RNA is transcribed is called the template strand. In some retroviruses that contain the RNA as the genetic material, example the HIV virus, the flow of information is reversed. The RNA synthesizes the DNA by reverse transcription, and this DNA that is formed is again transcribed into messenger RNA by transcription, and then this messenger RNA translates the proteins. Here you can see DNA by DNA polymerase undergoes replication to form DNA molecules. When DNA undergoes transcription, it forms the RNA by the enzyme RNA Polymerase. When the RNA forms DNA, that is called reverse transcription. The enzyme involved is reverse transcriptase. For a cell to operate, its genes must be expressed. This means that the gene products, whether proteins or RNA molecules, must be made. What is messenger RNA? The RNA that carries the genetic information encoding a protein from genes from the chromosome in the nucleus to the ribosomes that are present in the cytoplasm of the cell is known as the messenger RNA. The messenger RNA, the starting point of translation. DNA is transcribed into messenger RNA. Messenger RNA is translated into amino acids. Each codon, which consists of 3 nitrogenous bases, corresponds to 1 amino acid. Amino acids form the polypeptide chain. Polypeptide chains fold together to form the proteins. For a gene to be transcribed, the DNA, which is a double helix structure, must be pulled apart temporarily, and the RNA is synthesized by the enzyme DNA-dependent RNA polymerase. The enzyme binds to the DNA at the start of a gene and opens the double helix. Finally, RNA molecule is synthesized. The nucleotide sequence in the RNA complementary to the DNA template strand from which it is synthesized. Here you can see a double helix DNA structure which separates itself into 2 separate strands. One strand is called the template strand, the other strand is called the non-template strand. On the template strand, the complementary RNA strand is formed. Both the strands of the DNA are not copied during transcription. Why? If both the strands act as a template, they would code for RNA with different sequences. This in turn would code for proteins with different amino acid sequences. This would result in one segment of DNA coding for 2 different proteins. Hence, this can complicate the genetic information transfer machinery. If 2 RNA molecules were produced simultaneously, double-stranded RNA complement to each other would be formed. This would prevent the RNA from being translated into proteins. So let us know the differences between the replication and transcription. In replication, the DNA molecule separates into 2 strands in the formation of a replication fork. in the form of a Y, and one strand is called the leading strand, the other strand is called the lagging strand. The leading strand forms the continuous DNA molecule and the lagging strand forms the Okazaki fragments. Whereas in transcription, there is formation of a transcription bubble, and one strand is called the coding strand, the other strand is called the template strand, and the RNA is formed in the 5'3' direction in only one strand of DNA. Now let us move on to the transcription unit. A transcription unit consists of three regions: a promoter, the structural gene, and a terminator. What is a promoter? A promoter is located towards the 5' end of the coding strand. It is a DNA sequence that provides the binding set for the RNA polymerase. The presence of promoter in a transcription unit defines whether the strand is a template strand or the coding strand. The RNA polymerase binds to the promoter at the 5' end of the strand, and the strand unwounds, and the transcription of RNA occurs on the template strand. In eukaryotes, the promoter has AT-rich regions. These regions are called Tata box, otherwise Goldberg-Hognes box. And in prokaryote, this region is called Pribnow box. Besides promoter, eukaryotes also require an enhancer. Here you can see the prokaryotic promoter site where a DNA template strand is present. We have the Pribnow box, and next to the Pribnow box is the start of transcription. Below you can see the eukaryotic promoter site where the DNA template shows the CAAT box and the Tata box, Hognes box, and next to it is the transcription site. What is a terminator? The terminator is a region that is located at the 3' end of the coding strands. It contains a DNA sequence that causes the RNA polymerase to stop transcribing. Now coming to the structural gene. Two strands of DNA in a structural gene of a transcription unit has opposite polarity. One strand 3' to 5', the other strand 5' to 3'. Here in this image you can see the promoter site where the initiation of the transcription occurs. Next to it is the RNA coding sequence, and next to it is the terminator site where the transcription terminates. And the promoter, RNA coding sequence and the terminator together is called the gene. Now what is a template strand? DNA dependent RNA polymerase catalyzes the polymerization in one direction. The strand that has polarity 3' to 5' acts as a template strand. What is coding strand? The other strand, which has the polarity 5' to 3', has a sequence same as RNA except thymine instead of uracil and is displaced during transcription. This strand is called the coding strand. Here you can see a promoter site, a structural gene, and a terminator, a 3' to 5' strand called the template strand, and a 5' to 3' strand called the coding strand, and this is a transcription unit. Now coming to the structural gene in the prokaryotes and eukaryotes. In eukaryotes, the structural gene is monocystronic, whereas in prokaryote it is polycystronic. Monocystronic means each messenger RNA carries only a single gene and encodes information only for a single protein and is called monocystronic messenger RNA, that is commonly seen in eukaryotes. In prokaryotes, clusters of related genes known as operon, often found next to each other on the chromosome, are transcribed together to give a single messenger RNA, hence are polycystronic. Here you can see a prokaryotic messenger RNA which is polycystronic. Multiple translation sites are present and each protein is formed based on the particular code. In eukaryotic messenger RNA, single gene codes for a single protein, whereas in a prokaryotic, single gene can code for multiple proteins. or multiple genes can code for multiple proteins. Now coming to the RNA polymerase. Before starting transcription, RNA polymerase binds to the promoter site, which is a recognition sequence in front of the gene. Bacterial prokaryotic RNA polymerases consists of two major components, the core enzyme and the sigma subunit. The core enzyme is made up of 2 alpha, beta, beta to the power of 1 and omega. And this is responsible for the RNA synthesis, whereas a sigma subunit is responsible for recognition of the promoter. Here you can see the structure of the RNA polymerase, where you have 2 alpha, 1 beta and beta to the power of 1 and omega. The sigma protein recognizes the promoter and gets attached along with the RNA core enzyme to the promoter site. And the sigma remains at the promoter site and the core enzyme moves forward, adding nucleotides to the RNA chain. Promoter sequences vary in different organisms. RNA polymerase opens up the DNA to form a transcription bubble. Here you can see the growing RNA chain and the RNA polymerase being attached and the formation of the transcription bubble and the direction of synthesis from 5' to 3'. The core enzyme, as it moves ahead, manufactures RNA. It leaves the sigma subunit behind at the promoter region. Here you can see that the RNA polymerase recognizes the scanning site and the sigma gets attached to the promoter site and the core enzyme moves forward, adding nucleotides to the RNA and RNA chain is formed and the energy is released by the ribonucleotide triphosphates, where the energy is released from the 2 phosphates that is released, and at the end the sigma unit is released and the core enzyme moves forward. Now what is terminator? The end of a gene is marked by a terminator sequence that forms a hairpin structure in the RNA. The subclass of terminators require a protein, which is a recognition protein known as Rho, to function. Here you can see the Rho protein that helps in the termination of the RNA nucleotide chain, resulting in the formation of a hairpin terminating end. What is the process of transcription? In prokaryotes, there are 3 major types of RNA: messenger RNA, transfer RNA and ribosomal RNA. All 3 RNAs are needed to synthesize the protein in a cell. The messenger RNA provides a template, the transfer RNA brings the amino acids and reads the genetic code, and the ribosomal RNA plays a structural and catalytic role during translation. Now, the structures, messenger RNA is usually linear. It has a template to make proteins and carries the instructions for the polypeptide synthesis from the nucleus to the ribosomes in the cytoplasm. The ribosomal RNA makes up the ribosomes, which consists of a larger subunit and a smaller subunit, and forms an important part of both the subunits of the ribosomes. The transfer RNA is clover leaf shaped. It matches the amino acids to the messenger RNA to help the proteins formation. And the transfer RNA carries the amino acids to the ribosomes and matches them according to the coded messenger RNA message. There is a single DNA-dependent RNA polymerase that catalyzes the transcription of all types of RNA. How it initiated? It binds to the promoter and initiates the transcription. That is called initiation. The polymerase's binding sites are called the promoters. So the sigma factor and the core enzyme of the RNA polymerase gets attached to the promoter site. The sigma factor remains at the promoter site and the RNA polymerase moves forwards from the transcription start site. What is the substrate? It uses nucleoside triphosphate as the substrate and polymerases in a template dependent fashion following the rule of complementarity. So something about the initiation. The substrate for transcription for initiation is ribonucleoside triphosphate. that is added to the 3' end of the RNA molecule. Nucleotides are added one by one at a time to the 3' hydroxyl group of the growing RNA molecule. So 2 phosphate groups are cleaved from the incoming RNTP, and the remaining phosphate group participates in a phosphodiester bond that connects the nucleotide to the growing RNA molecule. Now coming to the elongation, after the initiation of the transcription, the polymerase continues to elongate the RNA, adding one nucleotide after another to the growing RNA chain. And the direction of synthesis is from 5' to 3' on the template strand. And the main enzyme required is RNA polymerase for the synthesis of the RNA. Only a short stretch of RNA remains bound to the enzyme. When the polymerase reaches a terminator at the end of the gene, the nascent RNA falls off. So also the RNA polymerase. Here you can see the RNA polymerase transcribes a region that forms a stem loop and then proceeds to the terminator. The stem loop causes the RNA polymerase to pause, and this pausing results in the formation of hairpin loop. During this pause, the Rho protein catches up to open the complex and separates the RNA and the DNA hybrid, and the RNA is separated and the RNA polymerase is also separated. What does the RNA polymerase do in the 3 steps? That is, first is initiation, second is elongation, third is termination. The RNA polymerase is only capable of catalyzing the process of elongation. The RNA polymerase associates transiently with the initiation factor sigma at the promoter site and the termination factor rho at the termination site. Association of RNA with these factors instructs the RNA polymerase either to initiate or terminate the process of transcription. Here you can see that the RNA polymerase, which consists of the sigma and the core enzyme, gets attached to the promoter site, and the sigma remains at the promoter site, and this initiates the RNA synthesis, and the core enzyme moves forward, continuing and elongating the synthesis of RNA strand, and when it reaches the terminator site, the termination occurs with the formation of a hairpin loop, and the Rho and the polymerase get separated from the synthesis strand. In bacteria, since the messenger RNA does not require any processing to become active, and also since translation and transcription takes place simultaneously in the same compartment, since there is no separation in the nucleus or the cytoplasm, so many times the translation can begin much before the messenger RNA is fully transcripted. This is because the genetic material is not separated from other organelles by a nuclear membrane. Consequently, translation and transcription can be coupled in bacteria. Here you can see the differences of the prokaryote and the eukaryote in the synthesis of RNA. The transcription and the translation occurs at the same time in the prokaryote, whereas in the case of eukaryotes, the transcription occurs within the nucleus. From the template DNA, there is formation of pre-messenger RNA and the formation of the messenger RNA, and this comes out of the nucleus and gets attached to the ribosomes and results in the translation of proteins, which occurs in the cytoplasm. In eukaryotes, there are at least 3 RNA polymerases in the nucleus, in addition to the polymerases found in the organelles. So there is a clear division of labor. The function of RNA polymerases: RNA polymerase 1 transcribes ribosomal RNAs, which are larger in size, like 28S, 18S, and 5.8S. The RNA polymerase 2 transcribes precursor of messenger RNA and hnRNA, heterogeneous nuclear RNA. The RNA polymerase 3 is responsible for transcription of transfer RNA, small 5S ribosomal RNA, and snRNA. Here you can see the differences. RNA polymerase is found in the nucleolus. It forms the larger ribosomal RNA, and the quantity present is 50 to 70%. RNA polymerase 2 is found in the nucleoplasm. It helps in the synthesis of H and RNA. The quantity present is 20 to 40%. RNA polymerase 3 is found in the nucleoplasm and helps in the synthesis of transfer RNA, and the amount available is around 10%. What are the differences between the prokaryotic RNA polymerase and the eukaryotic RNA polymerase? The RNA polymerase in prokaryotes consists of 4 subunits and a sigma factor. The 5 types of RNA polymerases occur in eukaryotes. The types of prokaryotic RNA, only single type. A single type of RNA is available in prokaryotes, whereas in eukaryotes, RNA polymerases are of 5 types: polymerase 1, 2, 3, 4 and 5. The core enzyme contains 5 subunits, whereas in the eukaryotes, it contains 10 to 20 subunits. The size is around 400 kDs, whereas the size in a eukaryote is around 500 kDs. Synthesizes polycystonic RNA, whereas eukaryotes synthesizes monocystonic RNA. Prokaryotes regulate transcription with the use of different types of sigma factors. Eukaryotic transcription is regulated by the presence of different types of RNA polymerases. Exons and introns. In eukaryotes, the monocystonic structural genes have interrupted coding sequence known as exons, express sequences, and introns, intervening sequences. Here you can see the introns and the exons. Exons are the coding sequences and the introns are the non-coding sequences. In a pre-messenger RNA, introns are present. In a mature messenger RNA, introns are removed. The removal of introns are done by a process called splicing. So, the splicing occurs by the spliceosomes in the presence of S and RNPs, and the excised introns are in the shape of lariat. And the mature RNA does not contain the introns, they contain only the exons. HNRNA undergoes additional processing called capping and tailing. In capping, an unusual nucleotide, methyl guanine triphosphate, is added at the 5' end, whereas adenyl residues 200 to 300 or polyadenyl residues are added at the 3' end in tailing. Here you can see the 5' end, there is methyl guanine, and at the 3' end there are addition of polyadenyl residues, resulting in formation of poly A tail. Thereafter, the processed HNRNA with removed introns and with 5' capping and 3' tailing is called messenger RNA and is transported out of the nucleus for translation. A mature messenger RNA will contain only exons. On either side, the 5' untranslated region is present. At the 3' there is untranslated region. At the 5' cap is present, and at 3' there is a poly A tail. The split gene feature of eukaryotic gene is almost entirely absent in prokaryotes. So originally, each exon may have coded for a single polypeptide chain with a specific function. So the polykaryotic gene, each gene can code for multiple proteins, whereas in a eukaryotic gene, single gene can code for single protein. And the prokaryotic gene does not contain exon or intron, it contains only coding region, whereas the eukaryotic gene contains intron and exons, and the introns are removed to form the coding region. Since exon arrangement and intron arrangement are flexible, the exon coding for these polypeptide subunits acts as domains combining in various ways to form new genes. Simple genes can produce different functional proteins by arranging their exons in several different ways through alternate splicing patterns, a mechanism known to play an important role in generating both protein and functional diversity in animals. Introns would have arisen before or after the evolution of eukaryotic gene. If introns arose late, how did they enter the eukaryotic gene? Introns are mobile DNA sequences that can splice themselves out of as well into specific target sites, acting like mobile transposon-like elements. And these elements stay inside the nucleus that mediate the transfer of genes between organisms. These are called horizontal gene transfer. What is horizontal gene transfer? It occurs between prokaryotic cells or from prokaryotic to eukaryotic cells and between eukaryotic cells. HGT is hypothesized to have played a major role in the evolution of life on Earth. Here in this image you can see that the horizontal gene transfer would have occurred from prokaryotes to archaebacteria or prokaryotes to eukaryotes, from plants to microbe to plants, between microbes. This has ecological implications in the evolution of biological species and in the endophytes. They also have a endophyte pyramid like the healthcare, bioactive metabolites, agriculture and plant growth promotion, environment phytoremediation. So with this we come to the end of this lecture on molecular genetics part 5. We have discussed about transcription. Thank you. Kindly subscribe, like, share and comment our channel readmedprep academy. Log on to www.readmedprepacademy.com. Our Facebook ID is readmedprep academy. Our email is readmedprepacademy@gmail.com. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.